As it became clear that the information of life was coded for in the DNA, that information must be passed on to offspring, work began to figure out how this could occur. Making a copy of DNA is known as replication. In fact, the word replication literally means to make an exact copy. The discovery of the structure of DNA quickly led to the realization of how easy it would be to make copies of it. With one side of the strand in hand, all that would be needed is to add the matching base, an A with a T or C with a G. We've already looked at the process of mitosis and meiosis to learn the mechanics of how the copies are distributed. Now we're going to look at the mechanisms of making an exact copy of DNA. It will involve three steps: unwinding, making copies, and tying up loose ends. One of the earliest questions asked is, what is the relationship between the new DNA strands and the original DNA strand? It's conceivable that you could use the original, keep it intact, and create a copy. This would be considered a conservative strategy, referred to as conservative because it would conserve the original piece of DNA completely. The other possibility would be that the two sides of the DNA strand would be separated, and then one new side would be created using the original as a template. This was semi-conservative because you would always have one original strand and one new strand, although the new strand would be exactly the same as the original. It was soon learned that the process of DNA replication was definitely a semi-conservative process. Knowing that it was a semi-conservative process and being aware of DNA structure, it was thought that a minimum of three steps must be accounted for in the process of making a DNA copy. First, the two strands must be physically separated and likely unwound from their helical shape. Second, free nucleotides must be added to the exposed sides of the base sequence. And third, any of the bases that are not joined together by covalent bonds on either side need to be joined by covalent bonds. DNA in its natural state is a twisted two-sided strand written in the nucleotides A, C, T, and G. Each base is joined to the adjacent bases using covalent bonds, but two bases from each side are held together by either two or three hydrogen bonds. The cell has within it an enzyme called DNA helicase. Helicase is an enzyme that undoes the double helix of DNA. It unwinds the twists, and in the process breaks the hydrogen bonds that hold the two sides together. Therefore, DNA helicase unzips and unwinds the two sides of a DNA strand. So as we continue, let's ignore the twists, the helices of DNA. If we look closely at a short sequence of DNA as part of a larger sequence, helicase has opened up the DNA and exposed each side. Now, a second enzyme called DNA polymerase comes into play. DNA polymerase looks at the exposed sides of DNA and picks the missing nucleotide to put on the opposite side. The issue becomes that DNA polymerase can only work in the 3 to 5 prime direction. As you recall, each strand is running in opposite directions. One strand, known as the leading strand, is in the correct direction, 3 to 5, and can be filled in as quickly as it's unwound. The other strand, called the lagging strand, is in the wrong direction. It runs 5 to 3. This strand must wait until enough of it is exposed for DNA polymerase to grab onto it and begin adding bases, thus leaving the lagging strand as a discontinuous segment of gapped sequences. After the action of helicase and DNA polymerase, we're left with two strands of DNA. One side is mostly complete, while the other side is made up of short segments called Okazaki fragments. These are named for the scientist who first described them. The Okazaki fragments and other gaps in covalent bonds between adjacent nucleotides are filled in and closed up by the enzyme DNA ligase. To ligate means to tie together, and that's just what this enzyme does, ties together any unbound nucleotides. So in the end, we have semi-conservatively made two copies of the original DNA strand. One side of each is an original strand, shown in black, while the other, shown in blue, is a brand new set of nucleotides. To accomplish this task, DNA helicase unzipped and unwound the original strand. DNA polymerase added bases in the 3 to 5 prime direction, giving us a leading, almost complete strand and a lagging with Okazaki fragment strand. And then DNA ligase comes and fills in the gaps and joins any unjoined adjacent bases. And that is semi-conservative replication in a nutshell.